rate of growth, normal cells. Cells can produce, but normal cells can stop this process once there are enough cells present. Usually once cells are contacted on all sides by other cells, they stop dividing. This is known as contact inhibition. Cancer cells. Unlike normal cells, cancer cells don't stop production once contacted by other cells, and they continue to produce. This causes them to pile on top of each other, forming a tumor. As cancer cells divide, a tumor will develop and grow. Cancer cells have the same needs as a normal cell. They need a blood supply to bring oxygen and nutrients to grow and survive. When a tumor is very small, it can easily grow, and it gets oxygen and nutrients from nearby blood vessels. But as a tumor grows, it needs more blood to bring oxygen to other nutrients to the cancer cells. So cancer cells send signals for tumor to make new blood vessels. This is called angiogenesis, and it is one of the reasons that tumors grow and get bigger.
Cell death. Normal cells. An important difference in the comparison of cancer cells versus normal cells is that normal cells self-destruct when they are diseased. This happens through a process called apoptosis, in which these cells break down and are eliminated by white blood cells. Cancer cells, cells do not have the ability to self-destruct, which is mainly because of the mutations that let the damage in the cells go undetected. Normal cells. The comparison of cancer cells versus normal cells shows that normal cells are assigned a specific task, and they always perform that task. For instance, the function of normal white blood cells is to fight off infections, and they do this all the time. Cancer cells. These cells are usually dysfunctional. The numbers of white blood cells increase exponentially in leukemia, but these cancerous white blood cells do not function properly, if at all. Normal cells. There are growth or tumor suppressors that control normal cells. But certain proteins handle this process, and these proteins are coded by various types of tumor suppressor genes. Some tell cells when to stop dividing, and others help fix damage in cells, and the last type takes care of the apoptosis. Cancer cells. These cells are a result of mutations in any of the growth or tumor suppressors that help regulate the functioning of normal cells. These mutations allow cancer cells to grow uncontrollably.
Cell reproduction. Normal cells, new cells replace dead cells or cells that became damaged. This reproduction is more important to maintain the cell population. Healthy cells reproductive properly by mitosis, except for sex cells that reproduce through mitosis. Cancer cells, when cells become cancerous, they begin to produce uncontrollably, mainly because they have chromosome mutations or gene mutations. They are in control of their growth signals and continue to multiply.
Cancer is typically attributed to hereditary and environmental factors, but these only explain a fraction of overall cancer risk. These factors fail to explain why some of the body's tissue types are more likely to develop cancer than others, in even cases where hereditary and environmental risks are equal. For example, tissues of the small and large intestine are both susceptible to cancer caused by mutations in the APC gene, which regulates the production of the APC protein, a tumor suppressor that keeps cells from dividing in an uncontrolled way. The small and large intestines are presumably exposed to the same environmental factors, and yet cancer of the colon, part of the large intestine, is far more common than cancer of the duodenum. In this study, researchers investigated how third factor chance genetic mutations that occur during stem cell division may contribute to the overall rate of cancer incidence in each tissue. Researchers plotted data from previously published studies to determine whether tissues with a greater number of stem cell divisions, and therefore a greater amount of DNA replication over a person's lifetime, is related to the overall risk of cancer in that tissue.